Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Sherry Dolan from Sartorius.
Hello, everybody. Thank you for the introduction, Leah. Happy Cinco de Mayo, everybody. Thank you very much for joining. I appreciate you taking the time today to listen to this webinar. So my name is Sherry Dolan, and I'm a technology consultant with Sartorius. The title of my talk is Comprehensive Virus Clearance Evaluation Using Small-Scale Membrane Adsorbers. So the agenda for this webinar is first, just a short introduction to using flow-through anion exchange chromatography for impurity and virus clearance. Next, I would like to present a case study that we recently completed with one of our customers. This customer was currently using membrane adsorbers in their processes. However, they did not have any experience using the Sartorius Sartobi membrane. So the purpose of the case study was to provide performance data to the customer so they could decide whether to put time and effort into further studies. And this case study consisted of two parts. The first part involved small-scale device comparison of virus retention using the two small-scale devices we have, the Nano and the Pico. The second part of the study involved a loading capacity study, where the goal was to determine the maximum amount of product that could be loaded onto the membrane adsorber in the flow-through mode before seeing virus breakthrough. Okay? And last, we will discuss why these loading studies are important, and we'll show a retrospective comparison of virus retention results. But before we get started, we would like to ask you a few questions. And I'll hand this over to Leah.
Great. So on your screen, you're going to see some questions. You can just take your mouse and go ahead and select the answer that works best for you. Then that would be great. So do you currently use anion exchange chromatography for impurity or virus removal? Yes or no? Have you used membrane absorbers for process development or clinical manufacturing? Yes or no? And have you used small-scale membrane adsorbers in virus clearance validation studies? Yes or no? So again, if you can just take your mouse and select the answer on each of those questions. I'm going to go ahead and give you a few seconds more to do that so that everyone in the audience can get it done. And then we'll turn it back over to Sherry. So let's just do a couple more seconds. If you haven't done so, please select your answers.
Great. So it looks like the majority of the audience does use anion exchange chromatography for impurity or virus removal. 64% of the audience uses membrane adsorbers for process development or clinical manufacturing, and less than 50% of the audience has used small-scale membrane adsorbers in virus clearance validation studies. That's interesting. So hopefully we can give you a little bit more insight on how to use these small-scale membrane adsorbers for these studies. Great. Thank you so much for answering the questions. So I will continue. You can still vote, but I will continue with the presentation. So anion exchange chromatography is commonly used in the flow-through mode to remove impurities and virus from the product of interest. In the flow-through mode, the goal is to have the product of interest positively charged, so it will flow through or not bind, right, to the membrane or the resin. And the impurities should be negatively charged, so they will bind to the membrane. And this is accomplished by choosing a process pH that is lower than the pI of the product and a pH that is higher than the pI of the impurity or the virus that you want to remove. The advantage of processing in the flow-through mode is that we can load high levels of product since the product is not binding. We can load high levels of product while capturing low levels of impurities and or virus. So membrane adsorbers are ideal for this application since they have very high flow rates, much higher than resins, and we're not diffusion limited. So viruses and many impurities have pI values less than 7, and most monoclonal antibodies have pI values above 7, so processing near neutral pH is common and effective. Just for your reference, the table on the right shows the PI value of commonly used viruses for virus removal studies. So here we see that MMV has two PI values. So a mutation in the wild type has resulted in a strain that has a PI of 4.5. However, the wild type has a PI of 6.2. And MMV is considered to be the worst case challenge for removal by anion exchange because of its high PI. And it's also small size, 20 to 25 nanometers. The small size can limit the surface charge that can bind to the membrane. It's also non-enveloped, which means it's a rigid virus, so that limits its ability to change shape and expose its binding sites. So MMV is considered worst case for anion exchanges, as I just mentioned. So these studies that we're going to present today show data for the MMV clearance. So our customer had three objectives. The first objective was to evaluate the Sartobine Q membrane adsorber as an alternate option in case of a supply chain issue with their current membrane adsorber. The second objective was to gather performance data for Sartobine Q. And this customer had no experience using the Sartobine, and a virus clearance capability was their main interest. The experimental study consisted of two parts. The first part was to determine the scalability of the Sartobine Q Pico to the nano device. These are the two small-scale devices. And the second part of the study consisted of a loading study where we took 4 different feed streams and we loaded the membrane to a maximum capacity of 20 kilograms per liter. We took fractions during the loading to determine at what point would the MMV virus breakthrough happen. That was the second part of the study, and we'll go through more details shortly. The last objective was to compare the MMV clearance on the Sartobine Q to historically generated data for their current platform membrane adsorber. So we're going to show the platform on this slide here. So purification platform processes are commonly used in the biopharmaceutical industry. So utilizing a platform process can significantly simplify process development. Our customer utilized the platform process shown here. So this process has the Q membrane adsorber placed as the second chromatography step after affinity capture and viral inactivation. So when the Q membrane is placed in this position, it's typically used to remove process impurities as well as virus. So we would expect to see a lower loading capacity on the membrane when it's positioned this way, since the impurities are going to bind to the membrane, and that's going to limit the amount of virus that can bind to the membrane. So placing the Q membrane, for instance, as a third chromatography step or as a polishing step where the feed stream has very little process impurities will likely result in higher loading capacities. So this platform process contains 3 orthogonal steps for envelope viruses. Those are the low pH inactivation, anion exchange for charge-based removal, and 20 nanometer filtration for size-based removal. And for non-envelope viruses, there are 2 orthogonal steps claimed, the anion exchange and the 20 nanometer filtration. Inactivation is only effective for the envelope viruses. So since there are only 2 virus clearance steps which are effective for the non-envelope viruses, such as MMV, it's really crucial to optimize each step to get maximum virus removal. So, and I'd also like to note here that often membrane adsorbers, which are used for early phase manufacturing, are replaced by chromatography resin for large-scale manufacturing. This is mainly due to economic reasons. So process developers will utilize the fast flow rates and the ease of setup for the membrane adsorbers, but often they don't use the membrane to its full capacity. Then when the bench scale processes are scaled up without this optimized capacity, the result is an expensive, non-feasible, large-scale membrane adsorber configuration. This non-optimized configuration is then often changed to chromatography resins. So by utilizing the high loading capacities, you can improve your economics and allow for continued use of the membrane adsorber from development all the way up to late-stage manufacturing. And we'll discuss this further in the presentation as well. So Sartorius offers 2 devices which are suitable for scaled-down models for virus clearance validation studies. So these devices are the Pico, which is 0.08 mils or 2.9 square centimeters, And we have the Nano, which is a 1 ml device, or 36 square centimeters. The Pico device is in a flat sheet disk format, and it has identical bed height to the process module. The Nano is constructed from a spiral wound membrane and also has identical bed height to the process module. So for our loading study, our aim was to load the membrane to a capacity of 20 kilograms of product per liter of membrane. And to do this, that would require 20 grams of product for each Nano run. So for the same loading capacity, the Pico would require only 1.6 grams for each run. So our customer had a limited amount of product available, so we offered to use the Pico device. And since the Pico device is very small, the customer did not have any prior experience with it, they asked us to perform the study to compare the performance of these two devices and show them that the virus retention capability will be the same for both devices. So again, thinking ahead to like a real late phase GLP study where you may be testing 4 viruses in duplicate, and potentially you may have a loading capacity of 20 kilograms per liter. If that were the case, the Nano would require 160 grams of product, which is a lot of product to remove from a GMP campaign to validate just one step in the manufacturing process. So in comparison, only 12.8 grams of material will be required if using the Pico. So for high loading capacity studies, the Pico really comes in handy and is really very useful. So for our case study part 1, we're going to discuss the scalability study. Here, as mentioned, the study purpose was to show that the Nano and Pico perform similarly and are interchangeable. The design of the study was to run duplicate runs with the Nano loaded to 5 kilograms per liter. A single run was performed with the Pico loaded similarly to 5 kilograms per liter. Our starting material was a material called bispecific 1, and it had a protein concentration of 20.45 grams per liter. Our buffer pH was 7.2, conductivity less than 5. Our flow rate was 10 membrane volutes per minute, and the flow through was collected as one pool, and it was analyzed for the presence of virus. So the results of the study. Here we see 4.76 and 5.48 LRV of MMV for the Nano device, and 5.11 LRV for the Pico. So this data confirms that the MMV, LRV for the Nano, and the Pico devices are comparable. So these results build confidence in using the smaller device for these high loading spiking studies. Now, I did want to note that the Pico device, it does require careful handling and full compliance with the user's instructions because it's due to its very small size, it's very important to fully wet the membrane, remove any air from the membrane, and protect the device from particles and or air bubbles entering the system. So failure to do so may result in pressure increases, and we have even noticed up to 90% decrease in binding capacity if the membrane is not fully wetted and air is not removed from the device. So we have an application note. It's called Using the Startobind Pico, and this application note provides detailed information on using these small-scale devices with a liquid chromatography system. So it's highly recommended if you want to try these small-scale devices, to please read our application note. Okay, case study part 2. So let's look at the case study part 2, the loading study test materials and buffers. So 4 different molecules, 4 different product types were chosen for this study to cover a diverse set of characteristics, and we'll show the characteristics in an upcoming slide. The test materials we have are called MAB, DVD, bispecific 1, and bispecific 2. The buffers we used were tris acetate, pH 7.2 with sodium chloride. The membranes were sanitized with sodium hydroxide. The device that was used was the Pico. And the test material was pre-filtered using a 0.2 micron pre-filter. We also had a 0.2 micron pre-filter placed in line of the Pico to protect it from any particles or aggregates that may form in the system, and also to work as an air bubble trap, to trap any air that may be coming through the system, because we don't want to get any air into this small device. The systems that were used for this study was the ActiPure chromatography system. So let's take a look at the characteristics of our test material. So in the upper left, we see the theoretical PI values for the materials. So as discussed earlier, we would like to have the pH of our buffer below the PI value of the product. So the product will be positively charged and will not bind to the membrane. As you can see here, 2 of our materials have PI values lower than our buffer. So our buffer is pH 7.2, and 2 of our materials have PI values of 6.9 and 6.8. So this means these products can potentially be negatively charged, hence they may bind to the membrane, which can interfere with the virus binding. Keep in mind, PI value is not the only factor that controls the charge of the molecule and whether or not it will bind to the membrane. The conformation of the molecule, the charge distribution on the surface of it can also influence the binding or whether it's not going to bind to the membrane. Anyway, looking at this, it's still kind of a red flag to have the PI value lower than the pH. Next, on the bottom left, we have sample pH. All of our samples have a pH greater than 7, which means the impurities that we want to remove should be negatively charged. That's good. The top right, we have our protein concentration. The protein concentrations range from 3.4 grams per liter up to 20.5 grams per liter. Protein concentration is not normally a critical parameter and should not have any effect on the virus binding. And last, on the bottom right, we have conductivity. So to achieve good binding of impurities and virus, when using Q chemistry, a conductivity less than 10 millisiemens per centimeter is needed. So here, our conductivities are all less than 5 millisiemens, which is really good. So now let's look at the impurity analytics. So we know that DNA and host cell protein should be negatively charged at pH 7.2 and should bind to the membrane. So here we see that all of our test materials contain low amounts of host cell proteins, with the exception of MAB and bispecific 2, which are slightly higher than the DVD and bispecific 1 molecules. Residual DNA level is below detection for all of the test materials. And the graph on the far right shows the high and low molecular weight species percent. So here, bispecific 2 really stands out as it contains a large amount of aggregates, 32%. So these aggregates may or may not be negatively charged at pH 7.2, and they may or may not bind to the membrane. Most aggregates are positively charged and will not bind to anion exchange media. In fact, cation exchange media is often more effective for binding of aggregates. However, there may be some acidic species of these aggregates that may bind to the membrane and affect the virus clearance. So moving on to the study design for part 2. The goal of the study is to determine the amount of test material that can be loaded on the membrane in the flow-through mode before the membrane becomes overloaded by our negatively charged impurities and virus breaks through into the filtrate. So again, we have our 4 different molecule product types. These molecules were sourced from their GMP campaigns as their anion exchange loads, their post-depth filtration. So here we wanted to fully challenge the membrane. Again, we loaded up to 20 kilograms of product per liter of membrane. Our conditions were the same as our scalability study with a pH 7.2 buffer, less than 5 millisiemens, at a flow rate 10 membrane volumes per minute. But here we took 5 fractions and we analyzed the fractions for the presence of the virus. So let's move on to the results. Here we see the results. We see that the MAB and the bispecific 1 showed minimal virus breakthrough all the way up to 20 kilograms per liter. On the right, we see a little different story. Bispecific 2 and DVD products show virus breakthrough after loading capacity of around 5 kilograms per liter. So the question is, why is there a difference with the loading capacity and virus retention between these molecules? Well, the simple answer is that there are negatively charged molecules binding to the membrane and competing with the virus for binding, especially with the bispecific 2 and DVD products. So it appears that the MAB and the bispecific 1 did not have any negatively charged molecules present, and the virus was able to completely bind to the membrane. So this retention profile is what we would expect for a feed stream that's very low in impurities, and usually this would occur when the starter bind is placed as a third chromatography step or a polishing step. We would expect to see very low impurities and very high loading on the membrane. So the retention profile for the bispecific 2 and the DVD is what we would expect when impurity removal is also expected from the membrane. So when the cytosine Q is placed as a second chromatography step, we expect to have lower loading capacities as there will be impurities binding to the membrane. So now, our next question is, which negatively charged molecules cause the virus breakthrough? And more importantly, can we predict the capacity based on the material attributes? Well, let's take a look at our material attributes again. So, what was binding to the membrane? Was it the product itself? Was it an aggregate, host cell protein, DNA? Could even have been endotoxin. We didn't mention endotoxin, but endotoxin will bind to Q membranes. So the MAB molecule showed complete virus retention all the way up to 20 kilograms per liter. And this is not unexpected as it has a high PI and it has low impurity levels. So that's not unexpected. Bispecific 1 also showed complete virus retention up to 20 kilograms per liter. So this is a little bit surprising to me, given the fact that the PI of the molecule is lower than the pH of the buffer. So there's a chance that the product could have bound to the membrane, limiting the virus that can bind. But however, this was not a problem because we managed to go up to 20 kilograms per liter without a problem. And the other impurity levels are low, so that shouldn't have affected the virus binding either. Moving on to bispecific 2, that molecule did show a decline of virus retention after 5 kilograms per liter loading. So this molecule also has a PI value lower than the buffer. And this molecule also has very high aggregate levels. So here, I would not suspect product binding since the bispecific 1 molecule did not bind to the membrane. I think a more likely suspect here is the amount of aggregate. I think there were species of aggregate that were negatively charged that were binding to the membrane. So last, the DVD molecule also showed a decrease in virus reduction after 5 kilograms per liter. So the PI of this molecule is high. The impurity levels are low. So I would really not expect to see a reduced capacity based on this information. However, an aggregate level 3% seems low, but there may be some negatively charged aggregate that is binding to the molecule, binding to the membrane, causing the decrease in virus retention. That is a possibility. So to provide further information, really an analysis of the product post-cytosine Q filtrate would help understand which molecules may have interfered with the virus binding. The analysis can possibly show a difference in aggregate level or host cell proteins before cytosine and after cytosine. However, keep in mind we are looking for a very small amount of impurities to bind, and it might be difficult to quantitatively measure this. So now I just wanted to quickly discuss product binding to the membrane. So when LRV values are lower than expected, product binding to the membrane is not always considered, and it should be. Small amounts of product binding can interfere with the virus binding. So for example, in the middle figure here, we see how little product is needed to fully saturate the binding sites on the membrane. So in our studies, we started with 160 milligrams of test material loaded onto the PICO. If the product was negatively charged and hence bound to the membrane, all it would take would be for 2.3 milligrams of protein to bind to the membrane to fully saturate the membrane. And the loss of 2.3 milligrams out of 160 milligrams of starting material calculates to be a loss of 1.4% product, or 98.6% recovery. So this could be difficult to quantitate this small loss, and you can see 99% recovery of your product, but still have product binding to the membrane. And looking at the graph on the left, here we see an example of binding to the cytosine Q membrane when the buffer pH is within 0.5 units of the PI of the product, and the conductivity of the buffer is low. So here we have an antibody with a PI of 8.5. When the buffer is at pH 8.5 and the NaCl concentration is low, below 100, we can see considerable binding to the membrane. So just to clarify the graph, the y-axis shows flow through relative to the max, which means product concentration of load divided by product concentration in the filtrate. So a value of 100% shows no binding and complete flow through the membrane. So increasing our NaCl or decreasing the pH to more than 0.5 units away from the PI will prevent most binding. So just keep in mind a small amount of product binding can affect our virus binding. So why are loading studies important? Here we show the impact of underloading the membrane during late phase manufacturing. In this example, we show the amount of membrane area required to process a 10,000 liter bioreactor with an output of 5 grams per liter. On the x-axis, we have loading capacity from 2 kilograms per liter up to 20, and on the y-axis, we have required membrane volume in liters. So just by increasing the loading on the membrane from 2 kilograms per liter to 5 kilograms per liter, we can see, 60% less surface area of membrane needed. And not only is, the membrane amount an important for the economics, also the buffer amount is much less. So we could save about 5,000 liters of buffer just by increasing the loading onto the membrane. So, but going further, if we increase the loading from 2 kilograms per liter to 20 kilograms per liter, that will result in a 90% savings on membrane area and a buffer savings over 7,000 liters. So by performing these loading studies and determining the maximum capacity, we can save many hundreds of thousands of dollars per year. And in addition, the smaller surface area requirement will result in a smaller footprint, hence a smaller pumps are needed and smaller skid size. So for the last part of the study, MMV retention results with Sartabine Q were compared to historical data for the competitor's product. So the MMV clearance for Sartabine Q was observed to be higher than the competitor for all 4 of the test materials. The load titers for these studies were similar for all of the runs. And here we see the LRVs ranging from 4.4 up to 5.6 with the Sartabine Q. So all of this work was performed at our virus validation lab in Glasgow. And my colleagues there have been working diligently the past year preparing the non-GLP lab for GLP certification. And they have a joint team experience of over 50 years. They've completed more than 700 studies. They have readily available study slots. They have highly purified viruses, which, by the way, it's very important for membrane adsorber validation studies to have highly purified viruses. And we have data to show that using an impure virus prep, that can negatively affect the virus retention. It's due to the impurities, the proteins and the DNAs that's in the virus prep can bind to the membrane and lower the capacity. So it's really important to use these highly purified preps. Our current virus panel consists of MMV, XMULV, Rio3, HSV1, and after the 3rd quarter of this year, we will have PRV, PPV, and BVDV. So just to summarize, the Sartabine Q Nano and Pico devices showed similar results, which supports the use of the Pico device for virus validation studies at high loading capacities. The maximum loading capacity will be impacted by the presence of negatively charged molecules that bind to the membrane in addition to the virus. So we clearly saw this in 2 out of the 4 molecules we tested. It's important to perform a loading study to determine the maximum loading capacity. Unfortunately, it's not possible to predict the maximum capacity based on the test material properties. As we saw in the study, we couldn't really pinpoint which attributes caused the breakthroughs. Sartabine Q membrane showed higher virus retention at similar load conditions when compared to the competitor's membrane. And the next step for the customer is to complete a cost analysis for using the membrane adsorbers at these high loading capacities versus the chromatography resin that they're using in late stage manufacturing. So finally, I'd like to acknowledge and give a big thank you to AbbVie, who collaborated with us on this study and provided the test materials, especially Richard and Kate. I'd like to thank all my Sartorius colleagues from the US and also from the Glasgow lab. And that is the end of my presentation. Thank you very much for taking the time to listen today, and I'd be happy to answer any questions that you may have.
Great. Thanks, Sherry. So we do have a few questions. As a reminder, you can go ahead and type in your questions, and even if we don't get to all the questions for the question and answer, we will be passing questions on to Sherry. So, can these small scale devices be used in bind and elute mode?
Uh, yeah, it's a very common question. And yes, the small devices can be used in bind and elute mode. However, we don't really recommend that the pico is used, especially for scaling, because you have to calculate the void volume in order to accurately calculate the capacity, and small errors in calculating the void volume will result in errors in the binding capacity, so that may cause problems with the scale up. So for scaling up, we would recommend use the nano for the bind and elute, and maybe for screening you could use the pico, but not for scaling.
Okay, and this is a clarification question. So what is your definition of breakthrough through the membrane?
Okay, so breakthrough happens when the virus does not bind to the membrane and comes out in the filtrate. So in the beginning, early on in the loading study, all the virus is bound to the membrane and there is no virus in the filtrate. And over time, the binding sites will become saturated and the virus will start to pass through the membrane with the product and go through to the filtrate. I hope that clears things up.
Yep. And how can pressure increase problem be prevented? How can the pressure increase problem be prevented?
So sometimes some customers do have problems with high pressures on, especially on the Pico device. So that's why it's really important to put a pre-filter in front of the Pico device to trap any air bubbles or particles or aggregates that might be forming. Sometimes when the product runs through the liquid chromatography system, it runs through the pumps and the valves and the mixers, that can cause aggregates to form. So having that pre-filter in line can trap all those large particles before they clog up the membrane adsorber. The membrane adsorber is a 3 to 5 micron pore size membrane, and we don't want to physically clog up the membrane. We want to utilize the charge and the electrostatic interaction with the products. We don't really want to clog up those pores because that would definitely affect our virus clearance.
And in terms of scale down model for the Pico and Nano, do you need to use different MV due to the dead volume that's different in different size of membrane absorbers, or how do you do the scale down model qualification?
Did you say MV?
That is what I'm seeing. Use a different MV due to the dead volume.
Membrane values, okay. No, we do a direct scale down. For the scale down, we really don't have to take into account the dead volume. We are loading based on grams of product per mil of resin, mil of membrane. So we do a direct scale down that way. So for flow through, it's a lot easier to scale down than for the bind and elute. The bind and elute, the void volume can complicate things, but for flow through, the void volume doesn't matter because you're loading a large amount of volume through, and the product is not binding, and you're loading a lot more product through. Bind and elute, you would be loading a smaller amount of product, so any change in the void volume will affect that small volume more than in the flow through mode. So to scale down, for the flow-through application, you just basically scale down based on grams of product per liter of membrane for either Nano or Pico. It's the same.
Okay. So it looks like we have time for just one more question. If we didn't get to your question, we will be passing those on to Sherry. And so go ahead and continue to type in any questions you have, and they will be passed along. But the last question for the webcast is, do you recommend to perform any type of membrane integrity testing prior to the use of the membrane?
That's a good one, yeah. So we do have an integrity test for these devices. And yes, we recommend that you perform a pre-use integrity test, especially on the Pico device. Now, the Pico device, due to its design, it's sensitive to pressure pulses. So I think the pressure pulse of greater than 80 some PSI can actually damage the Pico device. It's a little more sensitive than the Nano. So we do have an integrity test. It's a diffusion test that is performed, and it can detect damage that might be caused by pressure pulses, and it can detect bypass of the membrane. And you could do this integrity test before you use the membrane and after you use the membrane. But yes, we definitely recommend that you do the integrity test for the Pico before you use it. For the Nano, you can also perform the integrity test, but it's not as critical as it is with the Pico.
Okay. Well, thank you so much, Sherry. And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing us a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Bye. Have a good day.